Okay, hi everybody. Welcome to another session of Research Horizons Fall 2023 edition. Today, I have the pleasure of introducing Dr. Allison McDonald from Laurier University. She's a professor in the Department of Biology at Wilfrid Laurier. Her research investigates the mitochondrial enzyme alternative oxidase that is present in the electron transport systems of many organisms. Her lab uses an integrative and comparative approach to answer questions about bioenergetics in bacteria, fungi, plants, and animals. Dr. McDonald is an advocate for women in science and scientists with disabilities and strongly believes in the need for diversity, inclusion, equity, and accessibility in the scientific enterprise and our institutions. She regularly participates in training researchers in effective science communications, and her invited seminar today, which she has generously agreed to provide for us, is titled Flexibility in Making Life Work, Alternative Oxidase, and Building an Academic Career in Biology.
Wonderful. So thank you so much for the invitation. I'm really happy to be here today. My talk is going to be a combination of mostly research stories, but as part of those stories, I'm also going to provide, I guess, a little bit of advice or things that I've learned along the way in doing academic science in particular. I'm just trying to advance my slides here. Apologies. They seem to be stuck. Any suggestions, Valerio, to get around this issue?
Can you click on it and try to left, right arrow?
I guess we'll do it the old school way. We're going to go next.
Yeah.
So to start off, I do want to start with a land acknowledgement. So the city of Waterloo and Wilfrid Laurier University, where I live, work, and play, are located on the traditional territory of the Neutral, the Anishinaabe, and the Haudenosaunee peoples. And when I do these kinds of land acknowledgements, I do like to put a little bit of thought into it so that it's not performative. And so in particular, I want to call attention to some of the recommendations or calls to action in the Truth and Reconciliation report that have to do with education and educational spaces. And the first is the use of education as a means for starting reconciliation. And one of the first things that we can do here is, frankly, to educate ourselves so that when we are teaching, training, or mentoring others, we are doing that in a good, effective way. The second is to think about how in your practice you might want to integrate Indigenous knowledge and teaching methods into your classrooms or your laboratory spaces. So as an example, when I taught a third-year plant physiology course and we were talking about agricultural practices, I did mention what's called the Three Sisters, and this is Indigenous agriculture where we grow beans, corn, and squash together. And the plants not only support each other physically, but there are also elemental or nutrient interactions that are important there. So anywhere where we can talk about Indigenous knowledge in ways that are applicable to the courses that we're teaching, I think is always a great idea. And then lastly, to always think about building student capacity for intercultural understanding, empathy for understanding other people, as well as mutual respect within our spaces. So I just wanted to start my talk mentioning those things so that you can think about your own practices and what might work for you. This is difficult work. It's sometimes very uncomfortable work, but I will always make the argument that it's necessary work and that we should be doing it. So as a biologist and as a younger person, I've always been interested in organisms and how they eke out an existence, sometimes in situations that are very stressful, very extreme in some cases. So I was the kid that watched a lot of National Geographic specials. I watched a lot of nature things with David Suzuki. If you're familiar with other types of programming, you probably know David Attenborough, how he's always wandering into weird spaces to see what the organisms are doing in those spaces. And in particular, my key interest really is in metabolic pathways and the biochemistry that goes into being able to survive and thrive in what we would see as very extreme environments. So it's either really hot or it's really cold, or there's perhaps toxic chemicals or biochemicals in the environment. There are perhaps massive changes in pressure for organisms that are living very deep in the ocean, or organisms that are living up near the top of mountains. And then we also have some really interesting examples in biology where we have organisms either living on or within other organisms. And those are the environmental niches that they occupy. And so that's sort of how I really became interested in biology, was just these really neat organisms that live in places that we may not expect that life would be successful in those locations. And in my third year, I ended up taking an undergraduate cell biology course. And this is the organelle that I'm really into and interested in these days, and that's the mitochondria. So when you talk to either a high school student or an undergrad student, you say mitochondria, the first thing that you're going to get back from that person is they're going to say, ooh, the powerhouse of the cell. And it's kind of cheesy, but that is what they are known for. They are known for making copious amounts of ATP, which is the energetic currency that cells use on our planet. But mitochondria do lots of other things and are really cool in lots of other ways. So for example, they do contain their own genome, and I will probably come back to this a little bit later in my talk. They do a lot of cell signaling, in particular retrograde regulation, where they're talking to other organelles such as the nucleus or the chloroplast in plants. So they directly influence gene expression. And they also have other roles such as iron sulfur cluster assembly. And iron sulfur clusters are used in a lot of very ancient proteins that we all use in our bodies today that require that as a way for catalysis to actually work. We also know that mitochondria have a very strong role to play in the decision of cells as to whether they're going to continue living or whether they're going to undergo apoptosis or controlled cell death. And that's often turned on by the release of cytochrome C from the mitochondria. And then we're also starting to understand the key role that mitochondria play in many different types of human diseases. So often mitochondria, when they become dysfunctional, lead to a lot of unfortunate disease states in people. And then we also have a mitochondrial theory of aging, where as your mitochondria get older and their genome becomes more and more damaged, they don't work as well. And that's essentially why we grow old and eventually die. So I think mitochondria are really cool, and I got very interested in them as an undergraduate student. The other thing that I got exposed to in that third year cell biology class was this diagram. And this diagram used to keep me up at night because it was very terrifying, and there's a lot of things going on here, and it's pretty complicated. But now it keeps me up at night because it's really what I study, and I think it's really amazing how it works. So what I've put up here is a very basic schematic of looking inside a mammalian mitochondria. So to orient yourself, the cytoplasm is on the outside here. We have an outer mitochondrial membrane. We have an intramembrane space. We have the inner membrane. And then finally, we have the inside of the mitochondria, which is called the matrix. Now, this mammalian system is actually one of the most basic electron transport systems that we're going to see in any organism on the planet today. So you probably know that we can generate some ATP from glycolysis. We then move into the tricarboxylic acid cycle, where we make reducing equivalents. And those reducing equivalents are used by complexes 1 and 2 to pull electrons out. They're going to pass those electrons on to ubiquinone, which is a mobile carrier. It becomes reduced and then passes those electrons on to complex 3, cytochrome C, and then finally complex 4, or the cytochrome C oxidase. And those electrons are then used with oxygen and protons to make water. As those electrons are moving through this system, some of that free energy is actually used to pump protons from the matrix into the intramembrane space. And so what ends up happening is we end up with a huge number of protons in that intramembrane space, a very large gradient that develops, and then ATP synthase, or complex 5, uses those protons, basically to turn a molecular channel as they open it, and then we can convert ADP and inorganic phosphate into ATP. So this is a really amazing type of biological machinery. It works exceedingly well. It's very efficient, but there are a couple things that I do want to point out. So in our mammalian mitochondria, as I said, this system is very basic. So it's been stripped down over time, and these are the key components. It's fairly efficient. So most estimates that you're going to read in undergrad textbooks will tell you it makes 34 to 36 ATP as we move through these. And that's pretty cool because you're making a lot of energy. What is somewhat problematic is that this system is not particularly flexible. So for example, if an organism is in a particular environment and it runs into either a stress that's going to impact membrane fluidity, or it is exposed to some kind of inhibitor that another organism is making, these will often target this electron transport system. And if that happens and the system is inhibited, you have probably a couple of minutes to either move out of that environment and reduce your exposure, or you're actually going to die. So it's very efficient, but it's kind of putting all your eggs in one basket and hoping that you don't run into anything too problematic, because at that point, it may sometimes cease to work. So what I want to do now is contrast this sort of basic electron transport system with one that is a little bit more complicated. And I'm going to use the example from plant mitochondria. So this is very similar to the first diagram that I just showed you. The regular components are still here, but I do want to call your attention to two extra things. So at the front end, we have a series of what are called alternative NADH or NADPH dehydrogenases. So these are encoded by single genes in the nucleus. It's a single protein. And these are alternative ways for electrons to get into this system. So not just complex 1 and 2, but these alternative dehydrogenases as well. And in plants, there are actually 4 of them. 2 are on the outside or in the intermembrane space, and 2 are on the inside facing the matrix, which is kind of neat. So those can put electrons into the system. And in a similar fashion, we have a second enzyme that can take electrons out. So instead of using complex 3 and complex 4, we can use an enzyme called alternative oxidase in plants that takes electrons directly from ubiquinol and is also capable of connecting those with water and protons to make, oxygen and protons to make water. So there are a couple implications here. The first is the plant mitochondrial electron system is a lot more flexible than the one that we see in mammals, because there are additional ways for electrons to get in, and there are also additional ways for electrons to get out. So that's kind of interesting. One of the weird things about this system is if the plant is using alternative oxidase and the electrons are mostly going that way, then those electrons are actually bypassing or skipping complex 3 and complex 4. And that means that fewer protons are getting pumped across that inner membrane, which means that our proton motive force is quite a bit decreased. And that means that we're going to be making a lot less ATP. So from a bioenergetics point of view, that seems like a very unusual and perhaps disadvantaged choice that the plant has made through evolutionary time. So one of the key questions that I've always had is, well, what is going on with this alternative oxidase? It seems like a very strange little protein. I'm not sure why plants would have it, because it seems bioenergetically like a bad idea. What's going on here? And so that's sort of how I came into this field of research, is thinking about this funny little enzyme alternative oxidase and trying to determine, well, what is the deal with this protein? So the key question that I'm attempting to answer through my research program is, what is the physiological role of alternative oxidase? Obviously, it's not to make huge amounts of ATP. That is maybe a problem, but lots of organisms seem to have retained this protein over time. So I'm working on the assumption that it has some kind of physiological usefulness to organisms that have it. So we know one physiological role or function of alternative oxidase for sure, and that is in several groups that contain thermogenic plants. And so thermogenic plants essentially make heat, and it's a really interesting phenomenon. So what you're seeing here is a picture of what's called the corpse flower. And these do not bloom very often, but this is essentially the inflorescence of this organism. This one was taken in 2003. There are a couple people in this picture to give you a sense of the scale and the size of this inflorescence. It's usually between 2 to 3 meters tall by the time it's all said and done. And what's really interesting about this organism is it essentially channels all of its electrons during its reproduction phase, what you're seeing here, completely through alternative oxidase. And as a consequence of that, when you're operating and you're using a lot of alternative oxidase, the free energy change that's happening because you're not pumping protons has to be dissipated somehow. And what the plant does is it gives off that excess energy in the form of heat. So this central stalk that you're seeing, the spadix, really heats up, can get to ambient temperatures of 30 to 35 degrees Celsius, which I think is pretty impressive for a plant. As it heats up, it volatilizes a whole bunch of really interesting biochemical compounds, and it makes a really disgusting odor. So it is called the corpse flower for an obvious reason. It smells like dead, rotting flesh, which is not particularly appealing. What is neat about this is the pollinator in this system are carrion beetles. And so they are looking and smelling for dead corpses to go investigate and eat. And so the plant essentially tricks these beetles into coming to check it out. They crawl all up and down the spadix and collect pollen, and then they fertilize and complete the life cycle of this plant. So we know alternative oxidase in a subset of plants, those plants that are thermogenic, is used for that physiological purpose, for the purposes of reproduction. But it's a little confusing because not all plants are thermogenic, but we know that a good number of plants also contain alternative oxidase. So the heat dissipation is probably not always going to be the physiological role or the reason that an organism has alternative oxidase. So when I started my PhD work and started really thinking and investigating alternative oxidase, the situation that was happening at that time was a very focused approach to investigate this physiological role, as well as looking at how alternative oxidase is regulated. And the idea was that you'd study alternative oxidase in a lot of detail in one organism. You'd bring to bear a whole bunch of biological research tools to try and answer those questions. So things like molecular biology, biochemistry, and physiology. And this approach has been very successful. It started in about the 1970s and continues on to today. And it's given us a lot of detailed information about alternative oxidase, but particularly in flowering plants. And so the utility of this approach has been very good, but it's been somewhat limited because the focus has only been on a few organisms at a time. So the first lesson that I want to just impart here is when you're working in science and working in research, it's always good to have some kind of backup plan, because sometimes things don't always go how you think they're going to go. So when I started my PhD, the original plan A was for me to do a fair bit of investigation in tobacco. And I was going to use a particular vector to genetically engineer it. And that really was not working out for about 1.5 years. And that made me extremely nervous and very stressed. And so I decided, okay, if plan A never works out, I should probably be doing something else as a side project and start developing a plan B. And so that's what I did. And so my plan B ended up working with a lot of different organisms. So some diatoms, cyanobacteria, and then starting to use newer technologies such as bioinformatics to start asking questions about alternative oxidase. And so what ended up happening was plan A eventually really just got tossed because it never came to fruition for me. And what had started out as plan B actually turned into the major thrust of my PhD research, and later the various types of research that I've done as a faculty member. So when you can, it's always good to have a backup plan in place. So what I started doing was trying to take a more broad thinking approach to investigating this physiological role question. So what can we do to compare different things? So can we look at alternative oxidase in a wide variety of organisms, see what they're doing that is the same and see what they're doing that is different? And can that give us some hints as to the physiological role of this enzyme? At that time, which was the early 2000s, there were a lot of newer biology tools that were being used or starting to really come online to give us different types of information. So we had some sequence genomes happening in the late 90s. We were having people going into environments and just sequencing DNA. So we had metagenomics that was starting to come into databases. We had people starting to do similar types of things with proteins. So there was some new ways to start getting information, some new tools that could be brought to bear on this question. And then it might be useful to integrate the old and the new data together to try and generate, or perhaps to test hypotheses that we might have about alternative oxidase. So the first question that I've sort of tried to answer in the couple decades that I've spent studying this enzyme, and one of the first ones that I thought would be important to address is, what is the taxonomic distribution of alternative oxidase? And essentially that's just asking, what organisms even have this enzyme? Because we weren't too sure about that. And follow up to that, thinking about it a little bit more deeply, is, well, why would we want to do this? Why is it important to determine the organisms or how it's taxonomically distributed? So the first is, if we can figure out who has it, then we might be able to come up with a theory to explain why that is. So can we get around the question, or think about the question, of the origins of alternative oxidase and how it might have spread into the various organisms that do or do not have it? The other advantage that it could give us is it might identify some new model systems in which we can work. So I mentioned a lot of this early work had been done in flowering plants, but perhaps there were some other organisms that we might be able to work with as well. There were a lot of holes, I think, in the current knowledge, but expanding this taxonomic distribution information might really highlight where those gaps of knowledge were. They might also provide a really good context for what we already knew, but to highlight again what we're kind of not sure about. And then by knowing sort of who has it and getting a sense of, well, what does the sequence of the alternative oxidase look like in those organisms, it would give us a lot of sequence data that we could use for various types of analyses as well. So this to me seemed like a really good approach to essentially cast a really wide net and try and determine who has alternative oxidase. So when I sort of started my project, and this really got running in 2003, this is what we knew about alternative oxidase taxonomic distribution. So we knew a lot of plants had it, but most of the work had been done in flowering plants. So there's a fair bit of information from tomato, from tobacco, and from soybean, for example. We knew that some fungi had alternative oxidase, but it was pretty limited in terms of what had been looked at. And these were mostly human or plant pathogens. So C. albicans is a great example. And then we knew that some really interesting but terrifying protists also contained alternative oxidase. And these were mostly parasites of humans and animals. So things like trypanosomes, which cause sleeping sickness, or things like Cryptosporidium parvum, which causes a lot of gastrointestinal distress. So this is kind of where we were when I started this side project, and essentially went fishing and looking for alternative oxidase sequences, basically by teaching myself very basic bioinformatics approaches. And so by the end of that sort of chunk of time in my life, I've been able to determine that actually alternative oxidase has a really broad taxonomic distribution. So it wasn't just this funny little enzyme that only weird things had. It was actually in a lot of different organisms. So we were able to determine that alternative oxidase is actually present in all domains of life, with the exception of archaea. So that means it's in bacteria, and it's also in many different types of eukaryotic organisms. I had a lot of sequence data that I've been able to collect at that time, so there was a lot that I could do with that. And then for the first time, what was amazing and really fascinating and interesting was the first discovery of alternative oxidase present in prokaryotes, in bacteria. And so myself and then two other groups basically made that discovery around the same time. And that was cool because that made me feel good to know that that was a bona fide thing. And then a couple of years later, I made, I think, a very big discovery by showing that alternative oxidase is actually present in animals for the very first time. And this was a gigantic paradigm shift, because prior to that, what had always been written in the literature was animals do not have alternative oxidase. It's a thing that plants do, or fungus do, or protists do, but animals do not do this thing. So that's kind of a neat thing. So the second lesson that I want to impart here is, don't be afraid to challenge paradigms or rules or things that are supposed to be true in biology because it turns out sometimes they're not correct. So this challenging of what we think we know is actually how science advances, right? It builds upon knowledge. Sometimes we recognize that we've made mistakes and we back up a bit and we go and we fix it. And a really great example of this is the animal alternative oxidase discovery. So I had been searching these databases, pulling out sequences using bioinformatics. I was in the computer lab one day and I got an animal alternative oxidase sequence. And my first thought was, oh, this must be an error. This is a mistake. Because I had internalized the idea that animals don't have alternative oxidase. And I was very rigid in my thinking at that time. And then a couple of weeks later, I pulled out a second sequence from a different animal species and thought, oh, this is kind of strange. And then a couple of weeks later, alternative oxidases from 3 or 4 other different animals. And then I finally clued in and went, oh, well, perhaps I've discovered something really amazing and interesting. And I kind of had to let go that thought that animals don't have alternative oxidase, and perhaps that was incorrect. And it turned out it was, but it took me a little while to get there. So what I ended up doing was, as I said, finding alternative oxidase in sequences in 4 different animal species, and then trying to decide, okay, to really check. It's one thing to say that the sequence is there, but it's quite another to be able to demonstrate in a laboratory using experiments that this is actually correct. And that's what I felt I had to do. And so there were a couple different species, but a lot of them were really difficult to get my hands on. And I ultimately decided, okay, I can go with Pacific oyster. People eat them. I can find a place where I can purchase those. And that's what I did. So I couldn't find enough in the grocery store, and I ended up calling a wholesale seafood supplier in North York. And I drove up there one afternoon and I went in and I said, hi, I need 2 dozen oysters. And they're like, okay. And it was very sketchy. I walked in there, there was like these big plastic sheets hanging from the ceiling, these big containers with bubbling salt water. But it's the kind of thing that you see in, a police show where somebody walks in and they're going to be murdered in 5 minutes. So it was just a very strange experience. So I followed the guy to the back and he goes to the tank and he says, okay, how many would you like? And I said, well, I'd like about 24. And he's sort of fishing them out and putting them in a bag for me. And we get to talking and he's like, oh, do you work for a restaurant? And I said, no, because he's thinking I'm buying oysters because I'm going to sell them. And then he says, oh, well, are you having like a fancy party at your house? And I said, no. And then he turns to me and he says, well, what are you going to do with these? And I said, oh, well, I'm a scientist and I'm going to experiment on them. And he got really quiet and then he panicked and he said, these are clean. They don't have any parasites. And it turns out he actually thought I was doing a spot inspection of his foodstuffs because he thought I was with the Canadian Food Inspection Agency doing a surprise check. And I said, no, I'm a grad student. I'm actually just going to do experiments on these. And then he kind of calmed down, but it was a really bizarre, funny experience. So I took my oysters back to the lab and I just dissected out various tissues and body parts, which was pretty tricky because up until that point, I'd mostly worked in plants and yeast and not animals. And then I isolated total RNA from each of those tissues. And because I had a partial sequence from alternative oxidase, I designed primers to the sequence that I had. And then I did some reverse transcriptase PCR. And this is one of those amazing experiments where you do it and you don't have a lot of hope that it's really going to work out. And then you're amazed because the first time that you do it, it actually does work and it's very cool. So this is basically the results or the DNA gel of probably that very first experiment, which I then repeated a couple of times just to be sure. So on the left-hand side is a 100 base pair ladder. I was expecting a product of about 663 base pairs, and you can see in various tissues in the oyster, that's what I ended up getting. And then the kit that I was using, when you ran the positive control, you should see a product that's 323 base pairs, and that also worked. And then I didn't have anything in my negative control lane. So this was very exciting because it was really the first experimental proof that some animals do in fact contain an alternative oxidase. DNA at the genetic level, and then also able to demonstrate, yes, the transcript is there as well. And this was a big shift within the field of alternative oxidase because it basically destroyed that paradigm. So now we knew, yes, some animals do contain alternative oxidase. And more recently, we've sort of expanded the capabilities of bioinformatics analysis that we can do. And we've asked that question sort of, and our publication is just coming out this month. Myself and a collaborator, Ryan Weaver, use bioinformatics to really look at everything and to look at the presence or absence of alternative oxidase. So anything that's a white circle, these are groups that we have no evidence that alternative oxidase is there. So I already mentioned that archaea do not seem to have it. The other thing that I want to call your attention to is you and I, as we are vertebrates, also do not contain an alternative oxidase, which is kind of interesting. You can see though, when we look at plants, it's very widespread. Alternative oxidase is present in most of them. In fungus, it's kind of a mixed bag. We have some species in a particular clade that do have alternative oxidase, and then some that don't, which is kind of interesting. And then in this recent analysis, we did find some animals where we think there might be some horizontal gene transfer that's happened. So they have an alternative oxidase, but it doesn't appear to be an animal one. It appears to have been a fungal or a protist enzyme that somehow weaved its way into their genome. And that's very mysterious and is kind of a new thing. So that's something that we may follow up on a little bit later. So to have a closer look at what's going on in plants, I mentioned that a lot of the early work had been done in flowering plants or angiosperms. And then this is kind of filled in over the years as well. So I was asked to write a review paper over the summer, and that's just recently been published in Plant Phys. And it just sort of thinking about, okay, alternative oxidase in plants, what do we kind of know? And as a summary, it appears to be pretty ubiquitous. If we take any kind of plant, chances are it's going to have at least one copy of the alternative oxidase gene. There's a couple groups here, and I'll call your attention to the yellow stars. These are groups where we don't have any evidence that alternative oxidase is present, but I suspect that's just because the information on sequences is frankly not available in databases because there's a very small group of people that work on these plants, and there's just not a lot of molecular data available. But I can tell you that just last week, I was able to search in the ginkgo biloba genome and have found what I think is an alternative oxidase there. So I have a fourth-year student, and she's going to explore that in a little bit more detail this year. So this is really filled in since I started this work. We went from having a really good idea about it in angiosperms, and now really any type of land plant that you were to investigate, it's highly likely that you're going to find an alternative oxidase there. So that's sort of the first question, dealing with taxonomic distribution and what we think is kind of going on there. And we have a lot more information and data and a much bigger picture of the story now. So the second question is, well, now that we have a handle on who has alternative oxidase, can we come up for an explanation for where it came from and how it's spread to these many different groups of organisms? And that's really sort of question two. And we're kind of partway through this question. We have a good idea, but it's a little tricky to think about how to test this really effectively. So what we think is happening is, I mentioned that alternative oxidase was discovered in the mid-2000s by myself and a couple other people as being in bacteria. And our recent analysis has confirmed that it's pretty limited in bacteria and that it's present in proteobacteria. And if you've thought a little bit about or know some things about mitochondria, what you'll hopefully remember is that the closest living and thing that's alive today, closest related to mitochondria, are proteobacteria. And so that to me sort of started ringing alarm bells and put some things together. So I've sort of thought for a long time that alternative oxidase probably originated in proteobacteria many, many, many years ago, and that it has probably entered the eukaryotic lineage using endosymbiosis. And so this is the idea that we had two different cell types that were prokaryotic in origin. One is likely a proteobacteria. The other, there's lots of ideas about that partner. Many people think it was a type of archaea. And they come together, one cell engulfs the other. That's a process called endosymbiosis. And then out of that, we end up with a eukaryotic cell that contains a mitochondrion. And this is how I think alternative oxidase essentially got into the eukaryotic lineage, that it was originally a bacterial enzyme and it's moved in through primary endosymbiosis. Like I said, that one is a little tricky to test and to check, so we're still kind of wrestling with how to do that best. The third question, since we now have taxonomic distribution, we've sort of talked about where this thing came from and how it's spreading around, is, well, now that we have that information, are there different organisms that we can start looking in to ask questions or study alternative oxidase that are different than the ones that we've used in the past, because perhaps that comparative approach might be really useful. And so I'm just going to walk through this diagram, and it's a very much a schematic of evolutionary history on our planet. So if we start at the top, alternative oxidase is part of a group called di-RM carboxylate proteins. And these are very, very ancient proteins. They've been around for a very long period of time. We think that an ancient proteobacteria contained alternative oxidase because we have existing proteobacteria today that still contain it. And I just sort of told you the idea of the endosymbiosis side of things. So that primary endosymbiosis, we have a proteobacteria, maybe some kind of archaea, they come together, they form a primitive eukaryote with a mitochondrion. And off of that lineage, we have a lot of different organisms that we see on the planet today that we think originated there. So these are things like Jacobids, things like slime molds, fungi, animals, for example. And then a little bit later in evolutionary history, we think there was another primary endosymbiosis, and this is the one that involves cyanobacteria. And so we have a primitive eukaryote with a mitochondrion. It couples together with a cyanobacteria, and we end up with a protoalgae that has both a mitochondrion and a chloroplast. And from that organism, or group of organisms, there are three main lineages that come off of that: the glaucocystophytes, the red lineage, which leads to things like red algae, and then the green lineage, which leads to green algae, as well as all of the land plants that we're used to talking about today. And then things get very funky and interesting. So in addition to these primary endosymbiotic events between one prokaryote and another prokaryote coming together, a little bit later in time, we have what are called secondary endosymbiotic events, where we actually have a eukaryote and a eukaryote coming together. And this has happened in both the green lineage and the red lineage, and has led to a bunch of really interesting organisms on the planet today. Now, I put this up here just to call your attention to a couple of things. Anything or any group that's in a blue circle is where there'd been some investigation about alternative oxidase previously. So there'd been some work in apicomplexans, many of which are parasites of animals. There've been a lot of work on alternative oxidase in flowering plants. There've been some work in slime mold, some work in fungi, and then some work in green algae, such as Clematomonas reinhardti, and then a couple papers looking at euglena. Anything that's in a green circle was something that I identified as, hey, this is low-hanging fruit, probably somewhere where we should start asking questions or looking next. So maybe asking some of those questions about alternative oxidase in proteobacteria, perhaps starting to investigate it in animals, or looking at plants that are a little bit more basal. And so that's sort of what my group has started to do during the time that I'm at Laurier. The other thing as a grad student, when I was looking at this distribution that initially was very, very frustrating, was to figure out there were a lot of organisms still that did not contain alternative oxidase. And these were things like the yeast Saccharomyces cerevisiae and S. plombae, the bacteria E. coli, and then several animals such as fruit flies, C. elegans, rats, and people. And this was pretty disheartening at the beginning because all of these are really well-characterized model systems. They would have been great to work in, but unfortunately, none of them contained alternative oxidase. And so I wrestled and grappled with this for a really long period of time because it was very disappointing. But then I thought about what I'm going to tell you now, and that's lesson 3, and that's to think outside the box, to remember that science is about being really creative and coming up with neat, weird ideas and solving problems. And so what I did was I decided, well, I'm going to take lemons and I'm going to make lemonade. So I decided, well, I can take one of these model organisms that does not have an alternative oxidase, and I can use it as a way to heterologously express alternative oxidase from other organisms in it. And so that's what we did with some of our earlier animal work. We took the sequence from the Pacific oyster, we stuck it into Saccharomyces cerevisiae in an expression vector, And then we turned it on to sort of see what would happen. So we had 3 different strains of yeast with slightly different plasmids. The first plasmid just made beta-galactosidase. The second plasmid contained the oyster native alternative oxidase. We hadn't made any modifications. And then the last plasmid contained the oyster alternative oxidase, but I had removed the stop codon so that it would continue chugging into the plasmid a little bit, and it would add a multi-his tag on the C-terminal end of the protein. So now we have this tool that we can use to start asking some questions about oyster alternative oxidase. And we didn't have to grow oysters in the lab because that was not going to work. And so I just want to show you very quickly this Western blot. And what you're looking at here is on the left lane, we just have a protein size ladder. And then what we ended up doing was growing the yeast with our various vectors in the lab. And if they're grown in glucose, the system is not on. So they should not be expected to make alternative oxidase. When they're grown in the sugar galactose, that turns the vector on and they're starting to make alternative oxidase. And that's basically what we saw. So if they're in glucose, we don't see alternative oxidase. When we turn the system on and it's worked for a couple of hours, 4 to 8, then we do see the yeast making alternative oxidase. So this is a really nice system that we've used in the past. And I have a master's student and a 4th-year thesis student that are now going to put alternative oxidases from many different types of organisms into this system so that we can do some comparisons, some contrasting to look ideally at the post-translational regulation of each of these different proteins. And we can do it in a background that's exactly the same. The other thing that we did is with these various strains, we decided, okay, well, they're making the protein, but is the protein actually functional? Does it work? Is it a ubiquinol oxidase? So what we ended up doing was just a very quick plate growth experiment. So we put them on what's called non-fermentable media. And that means we're forcing them to have to use glycolysis, the TCA cycle, and the electron transport system. They can't use fermentation. We're going to force them to do this. And then what we did is we added various concentrations of cyanide to the plate. And cyanide inhibits cytochrome C oxidase. So it will completely poison and shut down that sort of mammalian electron transport system that I was talking about. But if the organism has an alternative oxidase, it's still going to keep respiring because that is still working. So we run a multitude of trials to get a handle on what was going on here. So I'm just going to show you a couple of plates. So on the left-hand side is a plate where there's no cyanide. So the yeast, regardless of what plasmid they have, should be growing quite happily. So on the left-hand side, this is the beta-galactosidase expressing yeast. In the middle is the histidine-tagged alternative oxidase from oyster. And on the right-hand side is the native alternative oxidase from oyster. And the top row, we haven't done any dilution of the yeast cells. The second row down, we've done a 10 times dilution. And the next row down, we've done 100 times dilution just to decrease the amount of yeast in each of those zones on the plate. So when there's no cyanide, everybody is happy and growing. But then when we added 2 millimolar cyanide to the plates, our beta-galactosidase strain, has some real problems. Our strain that has the alternative oxidase but with the his tag is pretty good, but does have some issues. And then our strain that's expressing our native oyster alternative oxidase has no problem dealing with the cyanide at all. So that to us is a good indication that not only is the enzyme being expressed, but it's actually getting targeted to mitochondria and it's actually working as it should. And so this is a system that we're going to, again, take advantage of this year and the next year and explore and express some other alternative oxidases from things other than this oyster. So lesson number 4, to sort of get into the last section of my talk, I always encourage people to not be afraid to work in a new organism. It's definitely more comfortable to stay with things that you're comfortable with and that you know really well. But by moving into new systems or new organisms, you're going to learn a ton. And so to always sort of keep that in mind. So for many years, we've done a lot of work in a lot of different things. We've dabbled in bacteria, we've dabbled in yeast, we've done lots of work in plants. And I wanted to come back to sort of this animal question again. And so I've been looking for many years for a decent system, a great type of animal that I could use to actually do some in vivo type work to investigate alternative oxidase. And several years ago, for the first time, I was able to identify that alternative oxidase is in a little organism called Tigreophis californicus. And this is a really small arthropod. It's a type of zooplankton that lives in salt water along the coast from British Columbia all the way down to California. And they have a couple of big advantages. So the first is they have a short generation time from egg to reproductive adult is only 3 weeks. They're pretty inexpensive to grow. They take up a very small amount of space because they're quite tiny. Their nuclear genome has been sequenced, so there's a little bit of tools that are starting to be developed there. And they're also very stress tolerant. And so because this question of environmental stress and alternative oxidase are kind of linked, I was looking for something where we could do this kind of work. And so we've done a number of experiments over the past couple of years just to get a sense of, are they making alternative oxidase? Is the protein being expressed? Are there certain types of environmental stresses that increase the expression? So we found a couple things where that is the case. So normally we grow these organisms at 15 degrees in an incubator in the lab. And under those conditions, you're looking at a Western blot here, looking for alternative oxidase enzyme. They don't really make a lot of alternative oxidase now. They've been acclimated for several years at 15 degrees. But if we do basically a 24-hour exposure and we shift them down to 6 degrees, or we shift them up to 28 degrees, then we start seeing the animals making alternative oxidase. And we assume this is in response to temperature stress, either cold or hot. And we saw the same kind of pattern if that exposure was a week long. So we think that alternative oxidase has some role to play in this particular animal in terms of dealing with environmental stresses. We've seen very similar things when we change the salt level that we grow the organisms in as well. So just sort of some take-home messages from this part of the talk. The first one is a little tongue-in-cheek. It's always work on something other than what your lab group's main focus is. And I kind of say that jokingly, but that's what I've ended up often falling into. And I've been able to learn a lot by doing that. It's certainly frustrating at times, but makes you more comfortable taking on sort of bigger challenges as you move through your career. I've also found that comparing and contrasting how different organisms do the same thing or similar things is very useful and can tell you a lot about different systems and how they work. And then perhaps most importantly, the big lesson that I've learned is to always keep an open mind and to not buy into the hype of biological rules or paradigms or certainties without really thinking about them very critically. Just because we have, a good number of papers that say that something is a certain way, probably, but there will always be exceptions to that. So it's always good to just not buy into it right away, to really think about these things carefully and critically before you just accept it. So coming back to our $1 million question, what is the physiological role of alternative oxidase? Well, we're starting to get there. We know thermogenesis is one of them. We strongly suspect it has a role in dealing with different types of environmental stresses in various types of organisms. So we're starting to narrow things down. But where my thinking is sort of at this point is that when we're talking about mitochondrial electron transport systems and bioenergetics in general, I think there is a real trade-off that's happening here. And I think the trade-off is happening between efficiency of electron transport systems and flexibility of electron transport systems. So as an example, if we think about that first electron transport system that I showed you, the one that is in our bodies because we are mammals, it's highly efficient. It probably is making large amounts of ATP. And because of that, we probably have a good amount of excess energy, a good amount of ATP that is available to do work. So not just maintain our bodies, but to do perhaps extra things. And over evolutionary time, perhaps this is translated into a really developed musculoskeletal system for locomotion, and a very developed brain and neurological network for sensing and interacting with the environment around us, because those things really cost a lot of ATP and are very expensive to maintain. We don't know the role that that's going to have in terms of, well, how long do these kind of organisms live? These mitochondria are working really hard for a long period of time. Many of you are probably aware that not all of those electrons stay in the chain. Sometimes they leak out, they find an oxygen, they're making reactive oxygen species, which are very damaging to our cells. And they cause a lot of degradation and breakdown, perhaps things like aging, perhaps things like mitochondrial disease. And then if your system is very efficient, that's great for making ATP, but do you have options when you run into a stressful situation or an inhibitor? Often you don't. You're going to have to locomote, physically move away from that stressor. And if that is not an option, you might be done. On the other side of things, we have things like plants that contain these alternative compounds or enzymes, such as alternative oxidase, and they have perhaps over evolutionary time decided on a strategy of flexibility. So perhaps there's less ATP being made because the proton pumping and the oxygen consumption are not directly tied or coupled to ATP synthesis all of the time. They're not as efficient, but there's a lot of flexibility in that system. There's multiple ways to move electrons through, and the system probably won't back up because there are those options. So if one system is inhibited, the other one can take over and vice versa. And again, what does this mean for lifespan effects? In terms of reactive oxygen species, if there's flexibility, we know from some data that ourselves and others have collected that the chain doesn't become as reduced. We have less electrons leaking out. We therefore have less reactive oxygen species being made. Perhaps that leads to longer lifespans. And as I said, there's probably some options there during stressful situations. If one system or the other is shut down, the other one is available as a backup, and perhaps that's good. And so we really have to think, I think, about bioenergetics because we've been very obsessed for a very long time that ATP is the only thing. It is the thing that matters. It's the key thing. And I will start making an argument that it is not the only thing. You can make tons and tons and tons of ATP, but if you run into a situation where your chain stops working, you're going to die. And I would argue that that is a worse outcome than perhaps being a little loosey-goosey and a little more flexible, and perhaps not as energetically efficient, but being able to survive these stressful environments or these insults or these inhibitors and getting by. I would argue that, being a little sloppy and alive is probably better than being dead if you're an organism. So some possible functions of alternative oxidase that we've sort of been looking into as well have other people in the field is originally, we probably think this enzyme evolved to essentially remove excess oxygen. So the Earth went from an environment that was very anoxic to an environment once photosynthesis, oxygenic photosynthesis got up and running, that suddenly there's this huge amount of oxygen that's been put into the environment and the atmosphere, and organisms had to deal with that. And at first it was probably very toxic. So having an enzyme that can convert oxygen into water, probably pretty advantageous. And then over time, we think that function has perhaps morphed into other things. So we think there's a role here for it to allow organisms to adapt to environmental stress. There's some metabolic flexibility in the mitochondrial electron transport system, and perhaps that provides some resistance to inhibitors or particular stressors. We also know that a lot of different pathogenic organisms or parasitic organisms contain alternative oxidase. And some research is now coming out about protists and fungi that are parasites that indicate that alternative oxidase is providing them some protection against reactive nitrogen species or reactive oxygen species that host defense immune systems are making. And so this could be very advantageous depending on the lifestyle of the organism that you're looking at. We suspect it's going to play a role in decisions about cell death or cell differentiation that individual cells are making. And we think that it might have some interesting roles depending on the life histories of some organisms. So as an example, if we're talking about plants, I've already mentioned the example of life history in some plants that are thermogenic, right? They fire everything through alternative oxidase. They're making vast amounts of heat. They release these volatile organic compounds. They are smelly. They attract insect pollinators. The plant gets pollinated. There are a couple other systems where we think there's some neat things going on. So another weird type of plant are carnivorous plants. These are plants that eat animals to get the nutrients that they require. We have some neat work being done by other people where they've seen that alternative oxidase use is increasing. Certain parts of these plants are warming up. Perhaps they're emitting carbon dioxide, which is a lure to lure in insect prey. For an example, a pitcher plant, and then the beetle lands, slides in, basically gets captured, and perhaps alternative oxidase might also be responsible for making lots of carbon skeletons, lots of acid to help perhaps with the digestion process. And then thirdly, lots of parasitic plants appear to contain alternative oxidase. We think there the increased heat role may have to do with those seed pods being able to pop open and those explosive discharges that we sometimes see. And a great example of this are various species of mistletoe, for example. So these are kind of some neat questions and some places that we might want to go in the future in plant biology when we're talking about and investigating alternative oxidase. So what is the significance of looking at alternative oxidase? Well, I always like to just do it because it's fun, it's interesting, it generates new information and new research directions. I think it can also provide us a better understanding of the evolutionary history of the enzyme. We have some pretty good ideas of what we think are happening, but again, testing those hypotheses are a little tricky. Many pathogens have alternative oxidase, you and I do not. So many people are starting to investigate alternative oxidase as a target for drug design to kill fungal pathogens and protistan pathogens in specific, because those are really hard to kill because they're also eukaryotic organisms. And then there's several groups in Europe that are looking at alternative oxidase as a potential gene therapy for certain types of mitochondrial diseases. So if you have a complex 3 or a complex 4 that is not working, usually due to some kind of genetic mutation, is it useful to put a second oxidase in front of those, bypass those complexes that are not working, prevent the backup of your electron transport system and the rest of your sugar metabolism to be able to live, right? So a lot of people with mitochondrial diseases, they manifest very early. They often die as quite young children because of the dysfunctions that their mitochondria have. A plug for my laboratory, I'll be looking for a postdoc in July 2024, and I will be also looking for MSc and or PhDs next September. So if you find alternative oxidase fascinating at this point, I would encourage you to please reach out because I'd be happy to talk to you about it in more detail. Some acknowledgements. So I've had a lot of great students and various trainees in my lab over the years. They have done the bulk of what I've talked about today. Laurier as an institute has been a great place to work. And I've also received various types of funding from various governmental agencies, which I really appreciate. And if you want to connect with me after this talk, here's where you can find me. So I'm on X, formerly Twitter. I have a blog that I maintain that I talk about, my research, but then also, as Valerio mentioned, I'm really interested in scientists with disabilities, women in science, various types of equity issues. And then that's also my email for where you can find me. So thank you very much for your time and your attention. And I'd be really delighted to take some questions if you have them. Thanks.
Let's thank Dr. McDonald virtually and in person here. Right, so at this point, I'll try to moderate some questions.
Awesome.
If you are online, just unmute yourself or raise your hand, or here if you have any questions, and then we'll try to. Okay, we got one here. Come up here, Jerome, to the mic. Where's the mic? Yeah.
Thank you for your talk.
Here.
Thank you for your talk. It was very interesting. I was wondering if you got some pushback from other scientists where you were questioning all this paradigm, just because like those questions or those assumptions were quite firmly ingrained, I think, in the mind by the people. So when you were questioning this and coming up with all this new information, you get some pushback or something like that.
Sorry, I missed half that question. I apologize.
Sorry about that. So what I said is, did you get some pushback from other scientists when you were questioning those paradigms?
Not a ton. most of the pushback came from myself. So things that I just internalized as, oh, well, this is true and this is false. And so kind of having to get over that. It was a little scary when I was a PhD student and used to working in plants, and I found alternative oxidase in animals. I made a choice to go to an animal biology meeting. And they were very friendly, but I was very intimidated because I thought, you know, I'm gonna give my talk and they're gonna tell me to get out of here because I'm talking about a plant enzyme, you know, but then in an animal system. And actually it was the complete opposite. They were really amazed and really interested. And that meeting is actually where I ended up meeting the person who would end up being my postdoctoral supervisor. So the power of networking is really important. So I haven't had, I think, a ton of pushback. I expect I'll probably get some more pushback on the whole efficiency versus flexibility argument, because many people who do bioenergetics, whether it's in mitochondria or chloroplasts, are very, very hung up on this idea of ATP being the only thing that matters. And, you know, the longer that I do this work, the more that I'm starting to disagree with that view.
Thank you. Lee-Marie.
Hi, thank you for the presentation. This might not be maybe the most relevant question in terms of what you presented about AOX, but I know that in animals, thermogenesis can occur by thermogenin in the mitochondrial membrane, which also results in an inefficient mitochondrial electron transport chain. So I was wondering if you have seen any correlation in having one over the other protein, or are there organisms that are known to have and utilize both mechanisms?
Yeah, so there's all of those different combinations. So when we look, so it's sort of called thermogenin. It's also considered uncoupling protein. And so when we look in animals, sometimes we have an animal that will just have an uncoupling protein, but it will not have an alternative oxidase. Sometimes we'll have one that just has an alternative oxidase, will not have an uncoupling protein. And then we have some where there's probably both present. I don't think anyone has done a very robust comparative analysis of who has which of these two things, and one or the other or both. So I would assume there's probably multiple different combinations. But what I can tell you is vertebrates don't appear to have an alternative oxidase, and many of those organisms do thermogenesis. So I have to assume that they're primarily massively relying on uncoupling protein instead. And so perhaps that's one of the choices that vertebrates have made, is they had these two things that uncoupled mitochondria. They didn't want to keep both running because perhaps that's too energetically costly, and they've picked one versus the other, and the uncoupling protein maybe won in that decision battle. But that's just a general hypothesis. I have no great evidence for that being the case.
Great, thank you.
I have a question.
Yeah.
So you mentioned, you know, the potential for, say, drug discovery, right, with alternative oxidase. I'm assuming, you know, you can take approaches like yeast to hybrids and things like that for using it as a way of discovering products that may be useful. But I was wondering, so just like people study mechanisms of human cells fighting tumorous cancer cells through mitigating ROS, is this possibly also a mechanism that plants have for mitigating tumors in plants? Or
Yeah, I don't know about the tumor question. They definitely use it for reactive oxygen species mitigation, for sure. I don't know enough about, you know, the only tumors in plants that I remotely know anything about are those that are caused by agrobacterium when we do transformation or out in nature as an infection. So it would be interesting to sort of look in those tissues and just see, you know, do we see ridiculous amounts of alternative oxidase, or is it somehow shut down and not working properly? And perhaps those pathogens take advantage of that. There's a couple of chemicals that we know target most alternative oxidases, and that's often what we use when we do respirometry to check. But there's also a lot of work happening right now looking at fungal alternative oxidases, because the coffee and chocolate industries have real problems right now with fungal pathogens taking out large amounts of their crops. And so there's a lot of desire to try and find compounds that can kill those organisms pretty effectively. And it is trickier because a fungus and a plant are both going to have alternative oxidases. So it's a question of, how precise can we be in our inhibition of one species alternative oxidase versus the other.
Interesting. Thank you.
You're welcome.
Okay. Any other questions? Moran?
Hi, yeah. So great presentation. Thank you so much, Allison. Yeah, I had a question like on the cost benefit of the function of this enzyme versus like a chemical quenching of reactive oxygen species.
Yeah, so the chemical quenching, I don't know how much of that is happening in mitochondria. There's probably some. I know there's a ton of that happening in the chloroplast of any plants. The other thing that's interesting is I've talked about alternative oxidase today, and that's a protein that hangs out in the mitochondria. There is an ortholog that we call PTOX, which is plastid terminal oxidase, and it basically has the same position, but in the photosynthetic electron transport system. And it seems to participate in relieving some of that over-excitation pressure that can sometimes develop in chloroplasts if we've got really high light levels or really ridiculous amounts of sugars that the plant is having to burn through somehow. Or we've got a massive over-reduction of that electron transport system. PTOX itself is really interesting because it's actually tangentially involved in carotenoid biosynthesis. So if you're going to make some of those quenching materials, you're going to be making some carotenoids or some xanthophylls and things like that. So a lot of that is directly kind of linked to each other. In terms of quenching in mitochondria, there's been a little bit of work, but it's decades behind kind of what we know about is happening in chloroplasts, for example.
Yeah, very cool. Yeah, thank you.
You're welcome.
Right. I think there are no further questions, seems like. So there are no questions. I will thank Dr. McDonald once again. And we hope to have you here in person sometime.
Thank you. I've appreciated it. Thank you very much for the invitation, and have a great rest of your week.
Yep, you take care.